Assalamualaikum warahmatullahi wabarakatuh and very good day. I am Madam Rose Azino binti Cik Lami. Today I'm going to give you a lecture on PhD 112 Cell Biology.
Oi, semua mari sini. Kau touch. Mari sini. Developer, kau pun mari sini. Cepat marilah. Berapa kali mau panggil lu orang?
Sorry, I terlambat sikit hari ni.
For the first lecture, I'm going to give you a talk about a basic cell features and prokaryotic cells. For the learning outcomes, at the end of the lecture, students should be able to explain the features of light and electron microscope. Students should be able to describe the characteristic of prokaryotic cells. For the introduction of cell biology, what is a cell? Cell is a microscopic mass of protoplasm, smallest living units of structure of organism. It is bounded by a membrane. It also contains a chemical active mixture of complex substances suspended in water. In multicellular organism, it is organized to form the structures and perform the function of that organism. What about the abilities of cells? Cells can survive on its own or has potential to do so due to hemostasis ability. It uses the enzyme and other proteins coded by DNA genes. It also has potential to reproduce by the cell division. It has sense and respond to environmental such as external or internal stimuli. For highly organized cell, it also responsible for metabolism. Components of all cells, it must have a plasma membrane or cell membrane. It also has a cytoplasm and organelles and also nucleus or nucleoid which contain the DNA. Why the cells aren't bigger? Because it is due to the surface to volume ratio. The bigger a cell is, the less surface area there is per unit volume. So above a certain size, material cannot move in or out of the cell fast enough. Now we talk about cells size and shape. The shape of cells are quite varied with some, such as neuron and also other cells. It is crucial factor in determining the cell's function. The size of cell is also related to their functions, such as egg or used in a Latin word ova is a very large, often being the largest cell an organism produces. The importance of cell because cell is the beginning of the other structure, such as tissue. Tissue is a group of similar cells that work together to perform the functions, while the organs is a structure and functional unit formed by various tissue that group together. Organ system is a group of organs that function together to carry out the principal activity of the organism. It is most complex level of organism and organism either multicellular or unicellular.
You faham tak?
Okay, this is the structure of the first, this is the replica actually of the first microscope. There are three persons that developing the cell theories. First is Amartya Sheldon. Second is the Theodore Swanch. Third is the Rudolf Virchow. Okay, the cells theory, which is cell is the smallest unit that having properties of life. Every organism is composed of one or more cells. All cells rise from the pre-existing cells. Okay, next we move to the microscope. Microscope can create detailed image of something that is otherwise too small to see using the naked eyes. currently we have two type of microscope. First is the light microscope, and also electron microscope. Light microscope is a simple or compound microscope, and second electron microscope, which is consists of transmission electron microscope and also scanning electron microscope. Here is the comparison of the size of the organism in Earth. Like a small molecules may below than 1 nanometer. Human, it could be from the 1 meter and up to the 10 meters. Limitation of light microscope. Wavelength of light are about 400 to 750 nanometer. The best light microscope can enlarge cells up to 2000 times, and light microscope can resolve objects down about 200 nanometer in size. Okay, this is the picture of the figure under the light microscope. Next, we move to the electron microscopy. We use stream of accelerated electron rather than light. Electrons are focused by magnet rather than glass lenses, and it can resolve structure down to 0.5 nanometer. Okay, this is the structure of the electron microscope. Okay, this is the real of the electron microscope. This is very big microscope. Okay, this is the figure that we can observe under the transmission electron micrograph with a thin section. Now we can see the differences if we use the same image for the light microscope. This is for the scanning electron micrograph. You can see a 3D. Here is the differences between the light microscope and also microscope. I hope you can read this later in your lecture notes.
Okay. Betul tak no? Okay.
Okay, all cells have a plasma membrane, enclosed cytoplasm and genetic material. There are two main types of cells based on organization of genetic material. First is prokaryotic cell, lacks a membrane-bounded nucleus. Second is a eukaryotic cell, which have a nucleus housing DNA. Now we move to the prokaryotic cells. Archaea and bacteria is a type of a prokaryotic cell, which DNA is not enclosed in the nucleus. It is generally the smallest and simplest cell. External features for the prokaryotic cell. It has a boundary which what we call as a plasma membrane. It also has a cell wall which consists of peptidoglycan and may secrete a slime or shell or capsule for protection. Some may have flagella or tiny projection called pili. The interior features of the prokaryotic cell, it has a single DNA molecule. It may have plasmid independent DNA fragment that carry specific pieces of genetic information. It also has a cytoplasm, and it has no internal membrane bounded structure. This means it has no organelles. The 70 subservage prokaryotic ribosome consists of 50 servage and also 30 servage subunit, as referred to a unit of density called a servage unit. Okay, this is the structure of the prokaryotic structure. It has a membrane, it has a cytoplasm with ribosome and also DNA openly DNA which is not enclosed with the membrane. It may also has pillars and also flagella. Alright, thank you for the lecture today. We will continue with the next lecture. Thank you.